So let's talk about the write-up of the paper, how you are going to change all these analysis into a proper research paper. So first of all, in the research paper, what you need the introduction. So in the introduction, you need 3 paragraphs. Sorry for that. So first paragraph should be about your disease that you are studying. So our is lung cancer. So we will write the paragraph on lung cancer. So it is according to your own need. If you want to, if you don't, you can change this. Second is about the bioinformatics techniques and why biomarker discovery is necessary. Why biomarkers are important. After that, we are going to the third paragraph, which will be showing us that what we are doing in our research, what we are going to do in our research. So after that, the introduction will be done. So how it will look like in your paper, I'm gonna show you a Milano cancer research paper that I was doing recently on the black tumor. So here you can see we have the Milano cancer research paper analysis, research paper write-up, where you can see introduction is written to the written to a proper paragraphing. So first paragraph is about cancer, second is about its development, third is about sorry for that. Third is about how to use the techniques and how they were treated recently. Fourth paragraph is about using the bioinformatics technique. And after that, we have moved to the material and method section. So now we are at the methodology. So 2 will be our methodology, first will be our introduction, and 2.1 will be the analysis that we perform. So first analysis we perform microarray data source of lung cancer. So how we took all this. So here you can see I have written this onto slides for you already. So if you change this to the a proper paragraph sample paragraph statements, you can see it's a proper write-up of the research paper. That's how you can write about write-up. So second will be already know that data processing. So we will paste that here, and then we will see if we have the data processing correctly done. So we will paste that here, and here you can see we have the methodology of performing the data analysis and data processing of differential expression. So now you are seeing we are getting the proper format. So then again we will move to 2.3, 2.2, 2.5 and things like that. After that you will be moved to the results section where your figures will be incorporated. So 2.1 will be the data source. Data source, what kind of data we chose. So what data set we chose, for that you can see in this quality control and data normalization. We have chose this data set, these much samples, and here are the distributions of the sample and their expression density. So we have 2.1 figure, 2.2 will be the DEGs identification, where we will put the information about our genes, differentially expressed genes that we have sorry about that we have identified. So we will move to our manuscript and paste the figure here, and above you can write that we have identified these genes, and that's this gene is involved in this one, the gene annotation that you got from gene card database, and then we will add the table in this section as well, that these are the DEGs that we have identified. Then you will move to 2.3, 2.4, 2.5 and things like that. So I'm just gonna show you here. So we have differentially expressed gene screening that we have done, and then we have the a protein and domain analysis that you can see here. After that, you can see the gene metabolite interaction network. You can do that as well. And then we have gene expression profiling, and you can do all the analysis by yourself if you want to. You can learn that on internet. Some that were very important I taught you as well. So after that, you will be moved to discussion. So how to write discussion? So discussion mainly consists of 1 to 2 pages. So discussion, and the first paragraph is about the study that you are currently performing. So we are studying the lung cancer and micro and biomarker discovery. The second paragraph will be about the study that you have done above. So we have identified biomarkers, and then in the third paragraph you will compare those to the previous results. So biomarkers that were identified previously, and now those biomarkers that are identified now that you have done. So that's how you will conclude this, and then you can see we will have the quality control and quality sampling. That here you can see we have the 2 paragraph discussion, and then we will conclude the analysis and conclude the research paper onto a conclusion section, and then your research paper will be done. After writing all this, you will have to write an abstract of the paper, that what you have done. So that is how I got these kinds of research paper done. So thank you for listening to this course and paying attention. If you want to, you can contact me anywhere to get the things done, get the analysis done, and get the guidance you want to do for the bioinformatics. And that is pretty much it for this course as well. If you want to, I will include some videos of how to add references as well, how to add a reference if you want to in your research paper, how to cite a paper using EndNote as well. So I will include that as well, and all the code that was used will be onto my GitHub repository. If by any chance I got missed that, you can message me. I will provide you the code, and thank you so much for attending this course. I am really thankful to you, and thank you so much for your support.